Okay, so let us start our meeting. So last week, we had a discussion about photosynthesis. And in our discussion, we were looking at the light-dependent stages of photosynthesis. And in that discussion, we were able to see what is called photophosphorylation. So I want us to start from there, recapping what we discussed. Then we can proceed to the dark stage or the light-independent stages of photosynthesis. So for the benefit of those who were not there, okay, it's not really for their benefit per se, but they are going to benefit because we are recapping. So those who were there, you can just be able to cross-check whether what you understood is what I'm putting forward. So basically, this is the schematic that shows all the two different types of photophosphorylation. It is showing us the non-cyclic as well as the cyclic photophosphorylation. So we have the non-cyclic, which goes like this, follow the cursor, like that, and then like that. So it is also sometimes called the Z scheme in some books because it follows the pattern of letter Z. And it's non-cyclic because the electrons are not moved around from where they started to back to where they were picked up from. So you find that the electrons are not cycled. The electrons never return to the place where they were lost from. So that's why it's non-cyclic. On the other hand, we have the cyclic photophosphorylation, which starts from pigment system 1 or photosystem 1 and goes like this to this electron acceptor, and the electrons are returned like this, and then they go back to the photosystem 1 or pigment system 1. So that is the non-cyclic, rather that's the cyclic photophosphorylation. And then when we are closing off, we talked about the differences between the two. And whenever you're dealing with differences between any physiological processes, it's very important that you have a criterion or you have a parameter that you base on to compare these two processes. Now, when we look at these two processes, we see that Wilson, help me mute these people. When we look at these two, we have to ask ourselves what parameters shall we use to compare. So one of them could be the number of photosystems or pigment systems involved. We could talk about the path taken by the electrons, the nature of that path taken by electrons. We could talk about the kinds of products that are produced. We could also talk about whether water is involved or not, and so on and so forth, okay? So how many electron acceptors are involved? So those are just hints for you to be able to make a good comparison. Remember, comparisons have to be about similarities and differences. So don't only look at the differences, but also consider the similarities. So in what way are these two systems similar? Okay, the similarity can be functional, it can be structural or otherwise. So that is food for thought. And I'll not do that for you. I want you to be very actively involved in your own learning. So I'm going to just run us through the step-by-step description, then we shall go to the dark stages. Okay. All right. So we start with the process of photolysis or photolysis. So here light energy is trapped in the pigment system 2, and then there's a boost of electrons to a high energy level. This is what I mean. So when these electrons are boosted to a high energy level, they are taken up by this electron acceptor, okay? And then we see that the electrons, okay, just mentioned that. Then we go to step 3, because these are my steps. This is step 1, step 2, this is step 3, then this is step 4, and we have step 5, okay, we have step 6, step 7, step 8, and step 9. Now, what is important is that what you are describing needs to make sense, okay? Needs to make logical sense. You don't just jump from here and then talk about what is here, then jump to the other part. Because this is a process, it means that things follow a chronological order or a sequential order. So that means for your things to make sense, you need to be very, very keen so that you don't lose marks. If your things are disjointed, then you find that you earn less marks than someone who has made the thing very orderly, okay? So that's why it's very important for you to understand the process, not to just know the events in the process, but understand the stages and why what one stage should happen before the other stage. So that is something to be keen about, other than cramming what has been provided. So I'm going to put some of you in the So I'm going to put some of you in the waiting room, those who are misbehaving. Okay, so let's move on. So we see that the third thing is that the electrons are passed from electron acceptor to another, to a series of electron carriers, and then to pigment system 1, or what we call photosystem 1, which now at this point is at a lower energy level. If you can just look at this. So we started by saying electrons are boosted to high energy level, taken up by electron acceptor. Then they are passed on to the electron carrier system. Now, the electron carrier system, they are later on taken by the pigment system 1 or photosystem 1. If you notice that this photosystem 1, pigment system 1, is now at a lower energy level than where they are coming from. Okay, so when it comes here, remember last time I told you that a photon of light strikes this, at the same time strikes this, at the same time strikes the water. So the striking of these 3 by photons of light causes them to lose electrons, okay? The water is going to be ionized or split to give electrons, hydrogen ions, as you can see here, and then oxygen given off as a byproduct. But then when these ones also lose electrons, then they are unstable, like I said. So the electrons coming from water will come and stabilize this photosystem 2, which had lost its electrons to a high energy level. And this one had also lost its electrons to a high energy level. So it is being stabilized by these electrons that moved right from here and went through all these stages. So that is something we need to take note of. So that's what I just said, that the electrons from the electron acceptor are passed to the electron carrier system and then picked up by the pigment system 1, which is at a lower energy level. Then we see that while that is happening, we are going to see that the energy lost by the electrons, because when an electron moves from high energy level to a lower energy level, it dissipates or it gives off energy. It loses energy. And remember, energy is neither created nor destroyed. Okay, so this energy is going to be picked up, okay, and utilized to convert ADP to ATP. It's going to convert ADP to ATP. And so we shall see that this is such an important step, because this ATP is later on put to use in the dark stage. So energy is thereby now converted to chemical energy. Remember, this energy was electrical energy. Now it has been converted to chemical energy. Okay, so let us proceed. If there are no questions, my class monitor should be able to guide me on what questions are being asked, as long as they are relevant to what we are discussing. So someone asked whether I'll share this thing on this video. No, I'm not going to share it. There's a recording which will be available on YouTube. So you should be able to go to YouTube and follow the discussion at your own pace. So I'm not going to share anything. So that is just what we say. The ADP is converted to ATP. Okay. ADP is converted to ATP. And that's what we call the phosphorylation. That's why we call it phosphorylation or phosphorylation. So phosphorylation, because a phosphate is being added to ADP to form ATP. Remember, this is adenosine diphosphate, then ATP is adenosine triphosphate. It means it's a nucleotide that has 3 phosphate groups. And this is a nucleotide. The adenosine is a nucleotide that has 2 phosphate groups. So the bond that is formed between these phosphate groups carries a lot of energy. So we call them high energy bonds. So these bonds constitute what we call the energy currency of the cell. So when a cell requires energy, it breaks these bonds, and the energy released is used to carry out the different, the energy released is used to carry out the different metabolic processes. So remember from your chemistry that that when you are forming a bond, energy is used, okay? And when you're breaking a bond, that same amount of energy used to form the bond is given out. So that is something that is very important for you to recall, okay? And this will be much more appreciated when you discuss the process of respiration. It is such a very important process. Okay. I think we can carry on. Gilson, do we have anything worthwhile in the chat?
So far, no question in the chat.
Okay. I hope people can hear me clearly. Kindly mute those people so that we don't have to put them in the waiting room. So whoever tries to mute to talk or to put on their video, please do the needful so that they do not disrupt the ongoing discussion.
Excuse me, teacher.
Yes, please.
Yeah, there is one has said, help me clarify more about ATP and DP.
ATP and
ADP.
Yeah, that's what I was just saying a little while. I'll repeat for their benefit. We know that cells or living things require energy, okay? They require energy to be able to carry out their metabolic processes. So what happens is that the energy is stored in a molecule called ATP. So this ATP is actually adenosine triphosphate. If you remember from your chemicals of life, you must have studied about nucleic acids, nucleotides, and the like. So among the nucleic acids, of course, we have DNA, which I think all of us should know by now, and then RNA. But those are not the only nucleotides. We also have ATP. We have NAD, NADP, FAD, acetyl coenzyme A. So all those are nucleotide, they have nucleotide molecules. Now, ATP is so important in every living thing because it's what we call the energy currency of the cell. Just to help us appreciate it, it's like if you have the US dollars, you can buy or sell in any part of this world, okay? So ATP is like that. It's universally utilized in every living thing. So all living things that when they require energy, they have to break the ATP molecule into ADP, okay? And the inorganic phosphate. But if they want to store energy, then they must form that molecule. Because again, from chemistry, we know that bond formation uses energy. Bond breaking releases energy. So at this point in time, we are storing energy, okay? We are storing energy, and this energy is got by the energy that the electron is losing. It is used to form a bond between ADP plus inorganic phosphate to form the ATP. So the third phosphate is called the inorganic phosphate. So when organisms eat foods that contain phosphate, then the phosphate is going to be used in this process. That's why you need to take seriously your vegetables and other such phosphorus containing foods, because they will come in handy. But now here, of course, the plants will have obtained the phosphates from the nutrients in the soil, and then they are used for this very purpose. Okay, I hope that is clear for the person who inquired about ATP. Okay, silence means they have nothing more to ask, I presume. So what happens is that the energy absorbed by the pigment system 1 or photosystem 1 is again given a boost to an even higher energy level, okay? We have a boost to an even higher energy level. So if you'll be able to see that shortly. They have a boost to an even higher energy level. That's this I wanted to draw this to your attention. And then what happens, so you see that here, this is at a higher energy level compared to here. Here is a lower energy level than here. So This was boosted to high energy level. Then it went down through the electron carrier system. And then now it was boosted to an even higher energy level. So that means it's going to have much energy within it, because each time an electron is breaks off from an atom, then it obtains a lot of energy and it's very unstable. Okay. Let's proceed. So that is what I've just said. And then the electrons are received by another electron acceptor. Remember, we said last time that if you know the names, make sure you talk about them, but they are not such a requirement because most of the books are silent about them. Only a few books talk about the identity. That is plastoquinone, plastocyanine, and ferredoxin. So in case you're not sure which one is where, you rather keep quiet. Only mention them when you are certain what they are, and they don't misallocate them, because they tell you lose a lot of marks. But if you know them and you're sure of what they are, then you can write them, but they are not such a requirement. So so that's what we just talked about. Then we see that the electrons which have been removed from chlorophyll are replaced by pulling in other electrons from the water molecule. That's what I talked about earlier. Okay, put this here. So this is what we are seeing right here. Remember that at the beginning, there were electrons that were lost from this photosystem. Okay, and now this photosystem is unstable. So for it to produce other electrons, they must replace what was taken away. So that is what we have there. Okay. And then we see the loss of electrons from the water molecule causes it dissociate into oxygen gas and protons. I think I talked about that earlier as well. Okay. So these protons are going to be used by this coenzyme. Okay. NADP. This is nicotinamide adenine dinucleotide phosphate. Okay. NADP. So this is also another molecule that contains nucleotides, but it's a coenzyme, and this is very important because it's going to accept these hydrogen ions. And from our chemistry, we know that when a substance receives hydrogen, we say it has been reduced. When it loses hydrogen, we say it has been oxidized. So that's why we call this reduced NADP. We usually read it as reduced NADP. So this reduced NADP is going to be very important together with this ATP. So these are the two very important products of the light dependent stages or the light stages. If these two products are not made, then you can never talk about the dark stage, because they are very instrumental in the dark stage. So that's why at some point I told you that whereas the dark stage can take place without light, okay, the dark stage will never happen unless the light stage has first happened, because without the light stage first happening and producing these things, then it means the dark stage has nothing to work with. So that is very, very important. It's a very important thing. So that's why when light is insufficient, it means these two products are not being made in sufficient quantities. And if they're not made in sufficient quantities, it means that the dark stage is going to occur very slowly, or it will quickly run out of what to use, and therefore less food will be made by the plant. Gilson, do you have anything in the chat?
So far, there is no one. or complaining or any message in the chat.
Okay, let's carry on.
Oh, excuse me, teacher.
Yes, please.
Yeah, the Emma is asking for a repetition. So may you repeat that side? I don't know which side is he asking.
Please, let's ask questions that are very clear to help me be able to help you soon and use less time. Okay. All right. If he cannot explain himself, then I can't be able to help. I see.
Sir, I wanted you just help me to repeat that part that you have just explained.
Okay, what I just said in a short while ago was the dark stage or the light independent stage of photosynthesis is very dependent. It relies so much on the light stage. So if in the light stage, if the light stage doesn't happen or it happens very slowly, then the dark stage will be severely impacted. Why? For two reasons: that very little ATP will be formed and very little reduced NADP will be formed. Two very important products that are going to be utilized in the dark stage. Okay, so that is what I said. All right, let's continue. So we see that the protons from the water molecule combine with electrons from the second electron acceptor, and these cause a reduction of NADP. Okay, what I told you that in chemistry, addition of hydrogen causes reduction. So the NADP will have an H on it, so we call it reduced NADP. And then we also see, okay, this is what I've just described, what we are seeing here, that the protons and these electrons together are able to cause the reduction of the NADP. Then we see also that the protons from the water combine with electrons. Okay, I just said this. Some of the electrons from the second acceptor may, okay, this is key, they may pass back to the chlorophyll molecule by the electron carrier system. Now here what we are trying to describe is the cyclic photophosphorylation. And while they are doing that, then they will yield ATP. So the plant has two options, okay? I would call them options, or it has two pathways. It can carry out both, okay? We are not really told when cyclic photophosphorylation occurs and when non-cyclic occurs. But what we know is that there's a possibility that both of them, either of them can happen, okay? So, but from what we have discussed, you can clearly see which of them is most beneficial for the plant, which of them is most beneficial for the plant and which one is least beneficial. But I want to think that this pathway doesn't exist for no reason. God has created it that way because of a particular reason. And maybe when you do biochemistry at a higher level, you should be able to find out what that reason is. Why is cyclic photophosphorylation there, yet it produces just ATP? There must be a reason. Okay. But for our level, we are not supposed to go into that detail. So here we see that the electrons from the chlorophyll are passed into the dark reaction via reduced NADP. So the electrons go into the dark reactions, but when they are part of this molecule. And these electrons are replaced from the water molecules without recycling back into the chlorophyll. So that's why we call it non-cyclic, because we see that the electron moved from here, then went here, then went there, then went there. It never returns. The electrons that are coming here are from water. You see, so the electron is going like that and never comes back. Unlike cyclic photophosphorylation, the electron moves up, then like that, and then comes back from right where it started from. So it's something that we need to be keen about. Okay. Okay, now let's do the dark stages of photosynthesis, or what we call the light independent stage. Now remember, this occurs in the stroma of the chloroplast. What we have been describing occurs on the thylakoids, on the grana. Now, we also know that this takes place whether light is present or not. Yes, Gilson?
Actually, it is, let me check here. He's asking, like, I would like to know the difference between, that is, I would like to know the difference between cyclic and non-cyclic photophosphorylation. That is coinus.
Okay, I'm not going to answer that. I'm just going to request 3 people to tell us the difference, because I think I have labored to draw that to your attention. I'm not going to answer that. Can 3 people give us the differences between cyclic and non-cyclic photophosphorylation? Any 3 people give us the differences. Someone is referring us to the page where it is in biological science, 206. If you have it, you can look there, but let's hear what people have to say. Dr. Pretty?
In non-cyclic phosphorylation photosystems, while cyclic involves only one photosystem.
Okay, that is one of them. Can we have other 2 differences? And by the way, those are not the only differences. Huh? So, Adams?
In cyclic phosphorylation, the first electron donor is the photosystem 1. Then in non-cyclic photophosphorylation, the first electron donor is water.
Okay, that is also correct. Let's have the 3rd person. Jesse?
In cyclic phosphorylation, photolysis
Jesse, you may consider typing your answer.
While in cyclic phosphorylation, photolysis of water occurs.
Okay, I've not heard you properly. Maybe you can type your answer. Let me ask someone else, since you have a network issue. Can someone explain what Jesse was saying or give us another answer? No answer, Dr. Pretty?
Chart, if we say that in non-cyclic phosphorylation, the products are ATP and reduced NADP, while in cyclic phosphorylation, the only product form is ATP.
Yeah, that is correct. I think we talked about that. Okay, I think those are just some. They're not the only ones. So I'm just going to leave it at that. So whoever asked a question, I think you are seeing the direction in which the answers are going. I started by saying when we had this discussion that ask yourself, what parameter am I comparing? Okay. The parameter could be products, the parameter could be process, the parameter could be, photo systems involved, and so on and so forth. Yeah, but if you look at BS 206, as someone guided us, you should be able to find the answers. Okot is saying that in cyclic photophosphorylation, there's no photolysis of water, while in uncyclic, yes, that is very true. Okay, that is very, very true. Okay, I think we move on. Now, this is an illustration of the dark stage. Now, the dark stage has also been called the Calvin cycle because of the work of Melvin Calvin. Melvin Calvin was someone who did a lot of research in photosynthesis and was able to come up with the processes, and he was honored by them calling it the Calvin cycle. Now, I want us to look very carefully at this thing. So we are going to start with stage 1. I think let me put this thing up so that we can, if I can rotate this on the side, because it's blocking some of the things. Okay, never mind. So what happens is that we have carbon dioxide. You see where my cursor is, my dot. So the carbon dioxide is going to diffuse into the stroma. Okay. When it diffuses into the stroma, it is going to react with a chemical compound known as ribulose bis-phosphate. It's a 5-carbon compound. And remember, carbon dioxide has only 1 carbon on it. Okay. So if you remember when you were discussing chemicals of life, you were taught about the types of monosaccharides. You were taught that monosaccharides are named according to the number of carbons that they have. The simplest monosaccharides are the trioses, okay, the triose sugars, where we had we had the glyceride and what was the other one? Glyceride is analdehyde. What ketose did you have?
It was dihydroxyacetone.
Yes, dihydroxyacetone and then glyceraldehyde, okay, as examples of the trioses. Then we say that there are also those that have 4, we call them tetroses. And then those that have 5 are called pentoses. Now the pentoses, we said we have ribulose, can underline it here. We have ribose, as in RNA, and then we have deoxyribose. I'm mentioning the ones which are more important, not that they are the only ones, but they are the ones which are important for us. So we have ribulose, we have deoxyribose, and then we have ribose. So the ribulose combines with two phosphate groups to form ribulose bisphosphate. Bis means two. Okay, so there are two phosphate groups joined on this ribulose. So this ribulose bisphosphate reacts with carbon dioxide to form a 6-carbon compound. Now, 5 plus 1 is equal to 6. So you get a 6-carbon compound. But what happens is that this 6-carbon compound is very unstable. Okay, it means that it does not last. Within a short time of its formation, it breaks down into 2. So if I divided 6 by 2, what do I get? If I divided 6 by 2, what do I get? Do we have people on this call? 6 divided by 2 is?
3.
Yes, it is 3. So if you divide this 6-carbon compound into 2 equal parts, you're going to get 2 carbon compounds that each have 3 carbon atoms. Okay? You're going to get 2 carbon compounds whereby each one of them has 3 carbon atoms. So that is now what we call glycerate 3-phosphate. That's what we call glycerate 3-phosphate. So what happens is that you have one glycerate 3-phosphate and another glycerate 3-phosphate. From your chemistry, the 3-phosphate is just telling us the position where the phosphate group has been attached on the glycerate. Okay? So some books don't say glycerate 3-phosphate, they just call it phosphoglycerate. Okay? Or others call it phosphoglyceric acid. So depending on which book you are looking at, you'll see those different alternatives, but they are meaning the same thing in essence. So it's important to know them because you never know, the question can be set with an alternative word. So these 2 molecules of glycerate 3-phosphate are now formed. So just for us to have some perspective, this is a carboxylic acid. That's why you hear us calling it phosphoglyceric acid. Or when you see the word ate, okay, like you can say oxalate, malate, okay, oxalic, okay. So this is a carboxylic acid. Now this glycerate 3-phosphate is going to now undergo an interesting reaction, okay? So the glycerate 3-phosphate is going to receive a phosphate group from this ATP. Remember that ATP comes from the light stage, okay? If you can see this here. So the ATP comes and then forfeits its phosphate group and then becomes ADP. So that means that now it is going to join onto this, okay? And then we are going to form what we call a triose or triose phosphate, okay? A triose phosphate. So we are going to form this triose phosphate. And what is also interesting is that at the same time, the reduced NADP, also from the light stage, is going to surrender its hydrogen. Let me maybe put the hydrogen here. So the hydrogen is going to come and join, okay, together with this phosphate, and then we form the triose phosphate. Now, some books call it the glyceraldehyde phosphate. Maybe let me annotate these words so that we can pick what I'm saying. So some books are going to call it pigal in short, which is, or others will call it gulp, which is a phosphoglyceraldehyde or glyceraldehyde phosphate. These are just short words to mean the same thing as triose phosphate. Biological science likes calling it a triose sugar or a triose phosphate. Okay. Then these GALP and PGAL are commonly used by functional approach, biology functional approach. So this one in that biology functional approach, they usually call this one PGA. PGA stroke GAP. Okay. So this PGA is phosphoglyceric acid or glyceraldehyde. Okay, mostly it is PGA. Let me remove this. So this PGA is phosphoglyceric acid. Those are just other terminologies that are used by different books. So what happens is that, of course, if the hydrogen has gone away, we are going to remain with oxidized NADP, oxidized NADP. So this oxidized NADP is going to go back into the light stage and wait to be given a hydrogen ion from the splitting of water, while this ADP is also going to go back with its, where it's going to go back and then wait for the addition of a phosphate, as we saw earlier. Okay. Now, what happens is that this triose phosphate or PGAL or GALP for that matter, they are carbonyl compounds. Remember, this is a carboxylic acid. Now, this is a carbonyl compound. From your organic chemistry, you know that you can reduce an acid, a carboxylic acid, into a carbonyl. In this case, our carbonyl is an aldehyde. Okay. So that is something to remind you from your organic chemistry. I don't know if you had covered that much, but if you have, or if you cover later, it will make much more sense. Then what happens is that this triose phosphate that has been formed, or PGAL or GALP, is either going to form hexose sugars, or most of it, and underline the word most of it is going to go back to regenerate ribulose bisphosphate. Ribulose bisphosphate is also called RUBIP. I think I had not talked about that. RUBIP. Okay. These books often call it RUBIP for short. Okay, can put this in capital letters too. They call it RUBIP, which is ribulose bisphosphate. Okay, ribulose bisphosphate. Of course, all these things are happening with the help of enzymes. Like here, there's an enzyme called ribulose bisphosphate carboxylase, because it helps the adding a carbon compound. So we call it a carboxylation reaction. So we find only a small portion of the TP or the GALP is used to form hexose sugars such as glucose. And then what happens is that the hexose sugars, of course, are going to be undergo polymerization to form starch, because starch is a polymer of thousands of hexose sugars, that is alpha glucose molecules. Okay, and then for the majority, or for most of the TP, it is converted into ribulose bisphosphate, so that the cycle can continue. Because if we do not form more ribulose bisphosphate, it means the one that is available will be soon used up, and then there will be no more ribulose bisphosphate to accept the carbon dioxide that is diffusing inside. So that is how this was designed. And then for that purpose, for this regeneration of ribulose bisphosphate, there must be energy involved. And this energy comes from the breakdown of the ATP molecule. So the ATP molecule is broken down into ADP, and the phosphates are used to regenerate the ribulose bisphosphate. So I think I'll first pause there because I think I have downloaded a lot on you at this point in time. So let me take questions and then we shall see how I can proceed from there. Are there any questions this far? Are there any questions
so far?
I'll ask again. Maybe some people have not had that question. Are there any questions? Yes, Adams.
Teacher, my question is that can proteins be also made in this cycle?
Proteins are made in a special process called protein synthesis. Now, if you remember, okay, that is something we are going to look at down the way. I am not going to describe protein synthesis, but I'll tell you how plants are able to make proteins. But in brief, what happens is that if you remember, proteins comprise of carbon, hydrogen, oxygen, and nitrogen elements. Now, insofar everything we have described here, there's no nitrogen involved. So the question is, how do proteins get formed if there's no nitrogen involved in photosynthesis? So photosynthesis is just about making carbohydrates, not making proteins. So, but the products of photosynthesis, some of the products of photosynthesis, or the intermediate compounds, let me just put it that way, the intermediate compounds are involved in the synthesis of amino acids, okay? So they are joined together with the amino group to form an amino acid, okay? And that amino acid is now joined to other amino acids during the process of protein synthesis, as it was described to you under, I think it was chemicals of life. So it's very important you bring those two topics together for you to get a good appreciation of what we are dealing with here. I hope, Adams, you have got a clear answer to your question. In case it's something that's not clear, then I would gladly elaborate.
Yes, teacher, I've understood, but what of lipids?
Lipids are also formed using the intermediates, some of these intermediates in the Calvin cycle, just like the proteins are formed using some of the intermediates of the Calvin cycle. But also there's another very important cycle you learn about, which is called the Krebs cycle, which is in respiration. So if it is those two cycles are very important in plants that result into the formation of proteins and lipids. And if time does allow, we shall be able to have a look at them, look at the illustrations that show how this comes about. Yes, Gilson.
Dr. Pitt is asking, how does a 3-carbon sugar become a 6-carbon sugar?
Now, this 3-carbon sugar, of course, you remember that there's a process called condensation. So small molecules join together to form larger molecules. So this 3-carbon sugar will undergo such a process to form a hexose sugar, which is a 6-carbon sugar, and then the 6 carbon sugars are going to again undergo other processes of condensation to form this polymer known as starch. And of course, there are other polymers like inulin, cellulose, and so on and so forth. All those are synthesized from the hexose sugar. So if the hexose sugar is beta glucose, then you think about cellulose. If the hexose sugar is alpha glucose, then think about starch, okay? And of course, you know of inulin and many others. So this is just to give you a brief description of what happens in the plant, okay? But otherwise, many more other compounds are formed. But for purposes of what I'm discussing now, we shall not go into that detail in how exactly they are formed. But this process is very important in their formation because the intermediates that are here are important starting materials for those chemicals to be formed. Do you have anything more? Okay, I'll repeat once, though more quickly, so that in case someone had lost a bit of network and they didn't get what I said, so that we are on the same page. So step 1 is carbon dioxide diffuses into the stroma, and it meets what we call a carbon dioxide acceptor molecule, which is rubip, okay? So this rubip is a 5 carbon sugar. So rubip will form a 6 carbon compound, which is very unstable, which immediately disintegrates to form 2 molecules. So I can say times 2. 2 molecules of 3 carbon compounds, which in this case are glycerol 3-phosphate or phosphoglyceric acid, PGA, okay? And then this PGA or glycerol 3-phosphate is going to undergo 2 simultaneous reactions. It is going to have a phosphate coming from ATP, so there's a dephosphorylation of ATP, and then we are also going to have a dehydrogenation of reduced NADP. So the NADP, reduced NADP is going to give up its hydrogen, while this one gives up its phosphate, and together they are going to form a phosphoglyceraldehyde, okay? Or glyceraldehyde 3-phosphate. So this phosphoglyceraldehyde or triose phosphate is going to now be used to form the hexose sugars. It is said as a small fraction, probably maybe 10 or 30% of it is used for the hexose sugars. So that most of the remaining triose phosphate molecules are going to be used to regenerate the rubip, this phosphate. And that process also takes energy, the ATP. So you can see that ATP is really very important. And if this ATP wasn't formed in the light stage, then it would definitely mean that there's no way the dark stages would take place. And so these hexose sugars that are formed will be polymerized to form starch and or maybe other polymers, like we said, cellulose and inulin and so on and so forth. So I think that in a nutshell is what happens during the dark stage or the Calvin cycle. Yes, Gilson.
There is a question from Konaise. He's saying, explain why when the plant is removed from light or exposed to light, the rubber levels gradually fall till they remain constant.
That's a very good question. Can you repeat it? Read it, say it slowly. Well, I think people can see it in the group. Okay. Can we all look at the chart and see the question before I attempt it? So this is a very, very good question. So asking us to explain why when a plant is removed from light, okay, or post-exposed to light, the rubip levels gradually fall till they remain constant. There are some questions that come like this, but in form of a graph, and you are expected to be able to explain the graph. So who wants to make an attempt at this question before I can bring in my contribution? Wants to? Adams?
For that question, I think since light is switched off, like it's put in the dark, that means that there is no formation of ATP during the photophosphorylation. And then ATP is needed to regenerate the ROP. So when there is no formation of ATP, that means there is no regeneration of the ROP. Therefore, it's going to gradually fall.
Okay, that is true. So when the ROP is not being regenerated, it means the one that is around is going to be utilized, okay, to form triose phosphate, and very soon there will be no more regeneration. And if there's no more regeneration, that means that its concentration will definitely have to come down. So that is a good answer. Okay, so let's move on. If there are no others, I think let's remove these annotations. Okay. So the overall process is like this: the reduction of carbon dioxide using reduced NADP and ATP. Carbon dioxide diffuses to the stroma of the chloroplast. Okay, so that is that. Then carbon dioxide combines with rubip bisphosphate, form an unstable 6 carbon compound. That one in most books, they don't show it. So I had to tell it to you so that you don't wonder how it comes about. Then 3, the 6 carbon breaks down into 2 molecules of glycerol 3-phosphate, GP. Some books call it GAP, others call it GP, others call it PG, but they mean the same. Then 4, ATP from the lysage will convert GP into triose phosphate. Then 5, the reduced NADP donates its hydrogen ion atoms to reduce the GP to triose phosphate, whereas this oxidized NAD will go back, NADP rather, will go back to the light stage to accept more hydrogen ions. That's what we've just said. Then we also see that the reduced NADP, this we just described, then the pair of triose phosphate molecules are combined, produce an intermediate hexose sugar. Okay, someone asked about that. Okay, and the hexose sugar is polymerized to form starch, which is stored by the plant. Okay. Then now we look at this stage where we have the regeneration of the ribulose bisphosphate. Now this is a combination of both. This is adopted from understanding biology. Here we have the light stage and the dark stage juxtaposed together. They have been put next to each other. So we have, let's start from here. The electrons, of course, in the cyclic photophosphorylation are moving in a cyclic manner. They are moving. So the chlorophyll molecules are excited, they lose the electrons to high energy levels, and they are taken up by electron acceptors, then they join the electron carrier system, then they return to the same chlorophyll. You see that? Then for the non-cyclic, what happens is that if this chlorophyll molecule, remember chlorophyll contains those photosystems. So the photosystems in the chlorophyll molecules lose electrons, and the electrons are going to be used to join to the protons that came from the splitting of the water. Then the electrons from the splitting of the water are going to come and stabilize this chlorophyll molecule. And as they are doing it, somewhere we are going to have ATP being generated. And then we see that these two protons are going to be used to form reduced NADP, which is going to come into the dark stage and cause the formation of a triose phosphate. Of course, the ATP that was generated from here, together with this reduced NADP, are responsible for the formation of a triose phosphate. Okay. And of course, before all that happened, it meant that carbon dioxide must have been absorbed and then accepted by ribulose bisphosphate and then formed into glycerate 3-phosphate, which now is able to be formed into a triose phosphate in the presence of ATP and the reduced NADP. And then the triose phosphate, some of it will form the starch, and the other is going to be just regenerate the ribulose bisphosphate. So if you look at this summary, it is emphasizing the substrates involved in the process of photosynthesis. We need sunlight. Sunlight is not really a substrate, but it's a condition. We need water for this sole purpose, for it to be split to release electrons and produce hydrogen ions. But in the process, we get a byproduct called oxygen. And then we see another important ingredient is the carbon dioxide, which is used in the dark stage. So we can see that of the two requirements for photosynthesis, water is used in the light stage, while carbon dioxide is used in the dark stage. I hope you can see that clearly. And then from water, we get two useful products. We get electrons and we get protons. And then we get a byproduct. Sometimes we can refer to it as a waste product, although in the true sense of the word, it's simply a byproduct because it can somehow be used also in respiration. Okay, because remember, respiration is a process that is always ongoing, regardless of whether there's light or there is no light. Okay, so it is a process ongoing, and so it can tap into some of this oxygen for it to occur. And then, of course, at the end of the dark stage, we have starch as our important product. So from the light stage, we have oxygen as a product, although we would better call it a byproduct rather than just calling it a product, because most of it diffuses out of the plant anyway. And that's how we are able to enjoy the oxygen that we have around us. Do you have any questions? Yes, Gilson.
There's a question from Ozzy. He's asking, why is it that when the concentration of carbon dioxide is high, the GP levels are high, yet those of RAP are low?
When the carbon dioxide concentration is high, the levels of RUP are?
The GP levels are high, yet those of RAP are low.
I'm not sure I've had that question. If it is a graph question, then it's important I know the graph, otherwise it may not add up. RUP concentration is high, less carbon dioxide binds to RUP. Someone is trying to answer or what? Okay. Why is root P high when GP is low? Yeah. Okay, can someone answer that? Single quote is trying to answer. Can someone make an attempt before we analyze Okot's answer? Why is it when this one is high, they're saying this one is low? I hope I got it right. Yes, Brian?
Teacher, I think like group B will only be high when GP is low, that's when there is no light. So there is an introduction of reduced NADP and the ATP to combine with yeah.
Yes.
When there is no light. So the GP now group P can be high when when GP is low when there is no light. So I got it mixed up. Let me first rethink again.
Okay. Gilson, you want to attempt?
So maybe I give it a try.
Yes.
The raw P levels can be high when GP levels are low due to factors such as limited availability of carbon dioxide. Now, if there is limited availability of carbon dioxide, it means that the rate at which raw P combines with carbon dioxide to form the 6-carbon compound is reduced. Now, this leads to an accumulation of raw P molecules in the system, resulting into higher levels of raw P. Yeah, maybe that one.
Okay, I think now that is much more clear and much better. Ozzy?
I think when carbon dioxide concentration is high, then the carbon dioxide combines with rubib, and then that makes it low. Since when it combines with rubib, the six carbon compound is split to make GP.
Not sure, Ozzy, you're answering the same question. But let's say the carbon dioxide concentration is low, then means rubib is sitting idle. Let me just use that language. There's less of it being converted into GP, okay? And therefore, if less of it is converted into GP, that means GP concentration is going to be low because what was there then had already been converted into these other molecules. You get it? So, and when we convert them into TP and the starch, and then more of it is, the majority is coming back and regenerating into rubib, means that the rubib concentration remains high. But if carbon dioxide concentration increases, then it means that more of the rubib is going to be converted into GP. And then the GP is going to be converted into these other things, okay? Of course, if there is no light, that means there's no ATP, and therefore it means that GP is not being converted into TP. And if GP is not converted into TP, it means now that GP instead will be the one to accumulate, and its concentration goes high. And then that of rubib will be going low because less of it has been regenerated. So it's that kind of thinking that you bring to this kind of question. You know, it is a cycle. So it means that if at some point of the chain there is stagnation, it means that other things downwards will severely be affected. And if they are severely affected, that means that their concentrations will go low, okay? And then the point where there was a disturbance, there will be a high concentration of that chemical because it's not being converted into the relevant compound. So if you have been in a place where there's traffic jam, assuming there's a junction, which is very important, say in the city, assuming maybe you are at Kampala Road and there is an accident, God forbid, and many cars are stuck in that jam, you will not be surprised to find that there are some places where now there are few cars because all the cars are stuck somewhere. Okay, so that is the thinking that works with this. Do you have any other questions? Okay, if there are no other questions, then I think it's better we go on as we still have some bit of time on us. So this is to answer what someone asked about proteins and lipids and what have you. So what is the fate of photosynthetic products? Do they just accumulate? What happens to them after photosynthesis has taken place? So we see that from the products of photosynthesis, these plants are able to make all the organic molecules they need to survive. Remember when we were discussing animals and plants in O level, we said that one of the things that make animals to have a lot of metabolic wastes is that they cannot synthesize their own compounds, so they must feed on others, and sometimes when they feed on them, they take more than they need, and then the excess is forms the metabolic waste. But when it comes to plants, they make just what they need. Everything they need, they can make for themselves, and they usually make just enough, okay? So if it is proteins, if it is lipids, if it is carbohydrates, they're able to make all those things. So let's start with the synthesis of other carbohydrates. The key word here is other, because we have been looking at the synthesis of starch, which is one of the carbohydrates. So yeah, what happens? Glucose and fructose combine to form sucrose. And this sucrose, if you can recall from your chemicals of life, is very important in plants. What is the role of sucrose molecule in plants? Why is it so important that they make sucrose? Why don't they just make starch and leave it at that? Adams?
Teacher, I think it's the form in which the organic materials are transported in a plant.
Okay. Thank you. Can we have any other answer? You on the track? Yes, so what happens when food has been formed? It cannot be allowed to accumulate in the leaves. It must be carried somewhere, okay? And it is carried in form of sucrose from what we call the source to the sink. Those who have studied transport, you know, there's a concept of the source and the sink. So the question is, why don't we transport this food in form of starch, or why don't we transport it in form of glucose itself? Because remember, we formed glucose at some point. Why opt for sucrose rather than the other carbohydrates?
Sucrose.
Continue.
Sucrose in most cases is not used by the plant during its processes of respiration and so it makes it so easy to be transported, and it's not used in the physiological processes of the plant.
That is one. But two, what do we know about sucrose in terms of solubility? Adams?
Sucrose is very highly soluble. which facilitates the generation of the pressure at the sink. Since water, there is more influx of water to generate that high pressure.
Okay. Yeah, so that is very important. So if you add the two answers, you'll know why sucrose is a choice carbohydrate. Then we can move on. Then glucose can be polymerized from starch. Of course, that one we've already talked about. Then also fructose can be polymerized to form inulin. Inulin is a type of polymer that is found in certain plants. I would challenge you, or I would task you to go on and find out some of those plants that have inulin. Okay, you'll be surprised that you have them in your compound, some of you. And then glucose may also polymerize to form cellulose, which of course will form the cell walls. Now, remember, this is beta glucose. Okay, this is beta glucose. These are alpha glucose. If you remember from your chemicals of life, okay, no, not this one. From your chemicals of life, you know that we have two isomers of glucose, alpha and beta glucose, which are different because of the position of the OH group on carbon number 1. Okay, one of them is down, the other one is up. And that small difference results in different molecules that are formed thereafter. Okay, and then we can look at synthesis of lipids. Now, here the glycerol 3-phosphate, remember the GP or the PG, which was formed after the dissociation or the breaking down of the 6 carbon compound that was unstable. So that some of the GP or PG is used to form a compound called acetyl coenzyme A. Acetyl coenzyme A is a 2-carbon compound. This one is a 3-carbon compound, which means for us to form this, we must lose a bit of carbon dioxide. So it's a decarboxylation reaction. Okay, so you will encounter this when you are discussing respiration. It's a very important thing. Remember, respiration is always taking place and it is being facilitated by some of these intermediate molecules that are formed during photosynthesis. So respiration is relying on them, okay, for it to form some of these compounds. And then we also see it can also be formed into fatty acids. Okay, from your chemicals of life, you know fatty acids are actually carboxylic acids with long hydrocarbon chains. Okay, and then we also see that the triose phosphate, okay, is going to form glycerol. Okay, now when we get a glycerol and a fatty acid, fatty acids rather, then you are going to have what we call lipids. Okay, if you remember from your chemicals of life, how many glycerols and how many fatty acids do we need to form lipids? Anybody wants to answer this? How many? If you know the answer, just interject.
Yes, fatty acids. 3 fatty acids.
3 fatty acid molecules with how many glycerol molecules?
1 glycerol molecule.
Okay, good. So you're going to form what we call a triglyceride, what do we call it? A triglyceride, a triglyceride molecule, or what we call a simple lipid. So this is a summary of all the alternative ways in which we can get these other molecules. So this part from here up to right, actually this is showing us a respiration pathway. So from up there up to here is what we call glycolysis or glycolysis, and that's what we call Krebs cycle. Now, you notice that when glucose has been formed from photosynthesis, some of it is going to be broken down to form a triose sugar, okay? And this triose sugar is going to later on be converted into pyruvate, okay? It's going to be converted into pyruvate. So what happens is that these processes are two-way. It's just that this arrow shows them as one way, but at the triose sugar level, we can form a glycerol, as I said earlier, or the molecules that go further to form a pyruvate and then acetyl coenzyme A, I told you there was a decarboxylation, they can be able to be joined. These are fragments which are two carbon compounds. Many of them are joined together to form a long chain of fatty acid molecules, okay? Long chains of fatty acid molecules. So these fatty acid molecules together with the glycerol molecules that have been formed, they can form the fats. Now, this is a two-way process. Let me just show you this. Depending the circumstances that the organism or the plant finds itself in. So if the plant wants to make lipids, it follows this pathway, okay? It follows this pathway. But in the event that the organism wants to metabolize the fats that it has, remember some of these plants store food in their seeds in form of lipids, like say castor oil, maybe simsim or sunflower and what have you, for the developing young plant. Then we are going to have that oil being broken down into glycerol and fatty acids, and then they are joining this pathway in this direction. Okay, so when you study about respiration, it will make sense. So some of these processes are two-way, and the direction of the process is dictated by the demands or the needs of the organism at hand. Yes, Adams?
And teacher, my question is that as I was looking through the mock papers, there is a certain question which said, describe the process of formation of lipids in plants.
Yes.
So am I supposed to start from the photophosphorylation if I'm to answer that question?
The question, it depends on how many marks and in what paper it has come from. Paper 1 or paper 2? Because
it was paper 20 marks.
Sorry?
Yeah, 20 marks. It
20?
was paper 2. Yeah, 20 marks.
Yes, that means now you have to write as many things as possible. That means you're going to show us the formation of the glucose, okay? And then you're going to show us how the glucose later on forms the triose sugar, and then how this triose sugar is going to be formed into glycerol. Then you're going to show us how the triose sugar some of it is used to form acetyl coenzyme A, and then how they undergo this condensation reactions to form fatty acids, and then they form the lipid. So you have a bit of that and you have a bit of this because you have 20 marks. If it was fewer marks, then you would just have a brief summary of the important stages. But because there are 20 marks, you have to write extensively about each one of those things that happens. So what happens is that for proteins, we have different types of amino acids. If you remember, 20 different amino acid types. Now, the amino acid types are divided into two broad categories. We have what we call the essential amino acids and the non-essential amino acids. Now, the essential amino acids cannot be synthesized by the cells, okay? Especially in animals. They must be taken in the diet. But the non-essential, they are synthesized from the essential ones, so a process called transamination. So that is what we need to take note of. So we see that to synthesize alanine, we must work with pyruvate. So the pyruvate will undergo certain processes which will result into the formation of an amino acid called alanine. So you're going to have, then you have, if you want to make glutamic acid or glutamate, then it's going to be synthesized from alpha ketoglutarate, okay? And then if you're going to synthesize aspartic acid or aspartate, it will come from oxaloacetate or oxaloacetic acid. So that is how the respiration results into the formation of some of these important compounds. So those are the things I've been talking about. So functions of lipids, I think those we know. I will not go so much into the explanation. So we can just read from the screen, but remember, these are not the only functions. There are many others. These are just a few of them. Then synthesis of proteins. So we see glycerol 3-phosphate playing a very important role, which results in formation of coenzyme A. And the coenzyme A can be very important in the Krebs cycle. And from the Krebs cycle, we can form other important amino acids. Then the non-essential ones are formed through transamination processes. Of course, like I said, at some point of our discussion, that form an amino acid, you must have nitrogen. So the nitrogen comes when the plants absorb nitrates from the soil, and then these nitrates are reduced into ammonium compounds, the ammonium group, then ammonium group, rather amino group. The amino group is now joined onto the carbohydrate structure or skeleton, and then we form the protein. So again, we are back at this. If you just follow the links. So we have a triose sugar forming a pyruvate and acetylcoenzyme A. Then it enters a Krebs cycle. Okay. Then we can get glutamic acid, or we can get aspartic acid, or we could get alanine. And then from this, we can synthesize non-essential amino acids. Remember, for children, there are 12, I think there are 12 essential amino acids. And for adults, there are 10 essential amino acids. At your level, you should be able to know what those 10 are, and essential amino acids, and what the non-essential are. The functions of proteins are provided there. Just a reminder. Okay. The class is quite quiet. I don't know. I hope people have not left their devices on. Are there any questions? What does glutamic acid do? Glutamic acid is a type of amino acid. And it's incidentally one of those essential amino acids. Glutamic acid is a soluble amino acid, which if there was a mutation, if you remember the case of sickle cell anemia, instead of forming glutamic acid, valine was formed, and valine is insoluble and resulted into the red blood cells crystallizing at low oxygen concentrations. So you can see how important some of these acids are. They determine the properties of the protein that is formed. So if there's a mutation and another amino acid is formed, then the properties of that protein will change and it will be very, very bad for the person that has it. Any other questions? So let's discuss factors affecting photosynthesis, because it's one of the important areas of photosynthesis. Can we mention factors likely to affect photosynthesis? Adams? Oh, let me see. Let's speak.
Light intensity.
Sorry?
Light intensity.
Light intensity. Edson?
Carbon dioxide concentration.
Carbon dioxide concentration. Gilson?
Chlorophyll concentration.
Chlorophyll concentration. Okay. What else? Is that all we have? So when you talk about factors that affect photosynthesis, you're going to consider external factors separately from internal factors. And now the external factors are the ones which we want to put our emphasis on, which include aspects like carbon dioxide concentration, light intensity, temperature. What else can we think about? Maybe availability of water, pollutants, and so on and so forth. Okay. But the most commonly discussed are those of light intensity, carbon dioxide concentration, and temperature. Okay. So we have the concept of limiting factors. This question has been asked before. What do we mean by the concept of limiting factors? Oh, oxygen concentration to some extent may affect the rate of photosynthesis, depending on the plant we are discussing. She's waiting, eh? Do you have anybody to explain the concept of limiting factors? Okay, so what happens here is that if there is a physiological process that is affected by more than one factor, okay? the rate at which that physiological process will happen is determined, or the speed at which it will happen is determined by that particular factor which is in shortest supply or which is at its minimum, okay? So, for example, let me just give you a situation which we probably are all familiar with. If you were constructing a house and you need a number of things to construct that house. You need workers, you need water, you need cement, you need bricks, okay, you need stones. Now, the rate at which that house will be constructed is determined by that factor which is in shortest supply, assuming you have most of the things, but you have 2 builders on site. So the speed at which the house will be constructed is dictated by the number of builders, since they are very few, okay? When you increase the number of builders, then the work goes faster. So it means that the builders were limiting the rate at which the work was going. But we can also flip the situation and say, assuming you have all the builders you need, but you have a handful bags of cement, maybe you have 10 bags of cement, everything else is in adequate supply. So you see that the work will stall or it will go slowly because you have very little cement. So the builders will tell you, we would have finished this work, but you see, you gave us little cement. So the same applies to photosynthesis, because many factors are involved in controlling the rate of photosynthesis. That one factor which is at its minimum value is the one which is going to cause the rate of photosynthesis to slow down. If it is light intensity, even if you have all the carbon dioxide necessary, you have the right temperature, but there's little light, then the rate of photosynthesis will be slowed down because of the sole reason that the light intensity isn't adequate. If the light intensity is adequate, but the temperature is low, and you have enough carbon dioxide, then the rate of reaction of photosynthesis will be slowed down. So that's what we call the concept of limiting factors. So if you change that limiting factor by increasing it, then the rate of photosynthesis will definitely increase. So this graph basically summarizes what I have been saying. And I think we shall stop on this graph. So we have light intensity is increasing from this point to this point. So you see at this point, the light intensity is very low. At this point, it has increased. At this point, it is moderately. And then at this point, it is high. And at this point, it is very high. And then here we are seeing the rate of photosynthesis. Now, we are doing this with low temperature, okay? And low concentration of carbon dioxide. When we raise the concentration of carbon dioxide, notice that the rate of photosynthesis increases even higher. Notice this value and this value, okay? We have increased the light intensity, but the rate of photosynthesis has remained constant simply because the carbon dioxide concentration is low. When we raise the carbon dioxide concentration, then you see that there is this increment, okay? There's that increment. And then when we increase the temperature to here, then you see that there is that increment. So that is what is very important when you're discussing such things. Now, in the interest of time, we shall stop here. And thank you for taking part on this call. I wish you all the best as you get back to school. I'd like to encourage you to read. And the reading I mean is not reading like a newspaper. As you are reading, you have to do active reading. ask yourself a number of questions and seek to find answers to those questions. So read while you're posing questions, and then those questions will be able to help you understand the subject matter. So we will stop here for now. Can we have someone lead us in a closing prayer? Yes, Ozzy?
Father, King of glory, we thank you for the gift of life. We thank you for our lesson. Father, we pray that you enable us understand whatever we are learning, and you also assist us as we are going back to school. Amen. In Jesus' name I pray. Amen.
Amen.